Hello, and welcome to It's Your Lymph, a podcast about all things immunity, health, beauty, and longevity. I'm your host, Lisa Levitt Gainsler, and I've dedicated my entire career to working with people's lymphatic systems because the results are nothing short of astonishing. Come along as we reconnect to the flow of life and learn how to harness the power of lymph. Hello, everybody. Welcome back to It's Your Lymph podcast. I am so excited for today's episode. I know you're going to be rewinding and taking notes during this episode. So let's jump in. My guest today is Dr. Sandro Demisquita. He is a leading researcher at Mayo Clinic's Department of Neuroscience in Jacksonville, Florida. His work centers around unraveling the role of the Meningeal lymphatic system, brain physiology, aging, and degeneration. Through his studies, he's provided unparalleled insights into the implications of impaired brain drainage by the Meningeal lymphatics, highlighting the interplay between brain function, behavior, neuroinflammation, and neurodegenerative diseases such as MS, Alzheimer's, Parkinson's, etc.
Thanks a lot for the kind introduction. It's a pleasure to be here. It was also a pleasure to bump into you in the elevator. It was very, very weird because you heard of my name and we never get the pleasure to chat before. But I'm really happy to be here and to discuss all things lymphatics with you.
Well, listen, the main reason why I started this podcast is that most people don't know much about their lymphatic system. And we're going to dig into, your path and how you got here. But really what I want is for people to understand more so they can really help live better lives. And if there's any role in prevention, where the research is going. Even the study of lymphatic science, lymphatic health is in its embryonic stage. So that's what I want us to do, is just help uncover some of these hidden messages that are in the bodies. But before we begin, I have what I like to call these icebreaker questions. So we get to know you and our audience gets to know you. So my first question for you is, what's your favorite place you've traveled?
So in my mother's side, we have very strong connection with France because of immigration back then. So there was I'm Portuguese, by the way. And there was a lot of immigration to France. And so my mother, she grew up in France. And so since I was little, I don't ask me to speak French though, because I learned it in school, but it's very rusty. Please don't. But so I'm getting there. So I really love Paris. I'm a big fan of Paris. I've been there several times. And I feel like every time I go there, there's this little, a glow about Paris in my head, and because I've been there since I was younger, and I have this connection, I guess. But the city is beautiful.
Beautiful. I love the way you describe that glow. Paris does have a glow. You're right. Okay, next question. This one I'm very curious about your answer to. If your lymph could talk, what would it say?
Ooh, stop eating. Stop eating all of those bad stuff. It's just go on a diet, I guess.
All right. Well, let's get into it. Let's get into that because later on in the episode, I do want to ask you about what things do you think are implicated in lymph that lymph does respond adversely to. But before we get that, I want the audience to hear about your backstory. Tell us about what you do in your lab, how you got to this career, really.
Well, it's a bit of a sum of different experiences and factors. Also, a lot of interesting and important people. Mostly mentors that I got throughout my career. But I would say, if I had to sum it all up in a word, most of it was serendipitous. Because I would say, throughout my, so I'm a biochemist by training, and when I was in college, I thought I wanted to go into food industry, right? Which is completely unrelated to what I'm doing now. But then I started having these experiences in the lab. I started doing, a bit more hardcore immunology using infectious models. And so that's when my taste for basic research started forming and then grow up. And then, again, a bit serendipitously, I ended up in a laboratory later on after I finished my master's that was doing neuroscience, right? And I said, and it's actually funny because I said, no, neuroscience, I'll never do that. I don't like it. I don't like the brain. And so, but then I kind of started liking it as well. I studied more and more. I started doing a lot of work related with neuroinflammation, especially in the context of aging and neurodegenerative disorders, as you mentioned during introduction. And I was studying also, so the brain is surrounded by this fluid. It's called cerebrospinal fluid. I was also studying how this fluid is formed and the composition of this fluid, how it's altered in disease context, and looking mostly at inflammatory mediators. And, I got to know, and this was during grad school, I was mostly focused on this. And then I got to know during one of these big conferences in the neuroscience field, I got to know Jonathan Kipnis, who later became my postdoctoral mentor. And so I did a big step in coming to the US because I did most of my training in Portugal. And this is really when I started to get a taste of top-notch neuroimmunology research and also the lymphatic system, because it was right at the time where the Yoni's initial study came out, which was really groundbreaking. It kind of opened a lot of eyes, and I basically I joined the team, and it's kind of history from there, right? I started working. I was very motivated. I think it's a fantastic field. We're still, as you mentioned as well, we're still trying to know more and more, and every time we take a little step, right, a bunch of other windows open, right? So it's, we're trying to figure out things little by little, and I think we're gonna get to a point where we're gonna find something that really makes a difference.
Thank you. I love that. And for the listener, Yoni is Dr. Kipnis. So
yes, it is.
just in case people were thrown by that. And, I mentioned Dr. Kipnis's work in my book, The Book of Lymph. Will you break down for our listeners the lymphatic drainage pattern of the brain and the cerebral spinal fluid and the, meningeal lymphatic pathways? So many people don't even know that. I mean, some of my listeners understand that the lymphatic system in the brain wasn't discovered really until 2011. But can you walk us through that physiology of brain lymphatics?
So I'm going to tell you about our current knowledge, right? Which is always something that is sort of mutating as we know more and more.
Oh, I have to pause on that line and highlight how important what you just said is. Our knowledge is mutating more and more.
Every time, right? And you have certain ideas that change when you have more facts. So it's always evolving, first of all, right? So what is really funny about this system, first of all, it's a lymphatic vasculature like the one, very similar to the one that you have in peripheral organs, right? So it's a continuation of our peripheral lymphatic system. It just reaches the borders of the brain. At that particular anatomical location, it has access to brain fluids and brain molecules, and also immune cells that are present at the borders of the brain. Okay, and basically we just didn't know it would reach that far, right? I mean, in simple words, it's not that it has this particular details and characteristics, because, for instance, and again, this is the facts that I'm going to mention is based on studies performed in animal models, okay, in rodents, right? So we're still trying to understand that what we're seeing in small mammalian systems, it's also applicable to humans, to us, right? We do know that they exist in humans because of imaging studies performed in non-human primates and in humans as well. But, in terms of morphology, cytoarchitecture, when it develops and matures, that's something that we're still trying to learn more and more. But what we know is that in small mammalians, in rodents, it develops postnatally, which is something that is really interesting because most of the lymphatic system in periphery develops in utero. So this aspect per se, right? It's already fascinating. What controls it, right? What leads to the development and the expansion of lymphatics into the borders of the brain and the spinal cord? We're still trying to understand, because maybe that also will shine some light on the same mechanisms that then lead to its degeneration during aging, right? Because during aging, what we know as well is that these vessels that drain the brain and spinal cord, they regress.
And they narrow, is what I read in the Kipnis research.
Yes. So the outcome is that they drain, they start to drain your less of your brain and spinal cord contents, okay? And we think that this is, again, having less lymphatic drainage is obviously, as you can imagine, is detrimental to any tissue.
And that brain inflammation, it is what's linked to those neurodegenerative diseases such as MS, Alzheimer's, Parkinson's, and more.
Absolutely. It's probably in those different diseases, it's a different flavor of inflammation, right? So it's not like you have the same inflammatory response across the different diseases. No, each disease has their different components. And I think right now what we're trying to understand is how the lymphatic system changes in all of those contexts, and how does it contribute to these different flavors of inflammation that then potentially we can actually harness the lymphatic system to correct that and to foster proper brain function during aging process. And this is kind of at the stage that we are right now. Okay,
so question for you. When I heard you gave a talk at the Lymphatic Forum in Banff, which was awesome, and I wrote like all these notes, and I was like, oh, I can't wait to have them on. So I have you again, just like the elevator captive. And what I want people to understand, what I want to understand is some of what we had previously thought about lymphatic clearance in the brain happens when we sleep, like the cerebral spinal fluid. But from your talk, that's when you're talking about the different behaviors of how lymph works in the brain. I imagine lymph is draining in the brain while we're awake. But can you speak to waste clearance and when it occurs?
Yes, it's something that is under development, right? All of these concepts. Because, now, so what you're referring to is at the level of the brain itself, right? The brain tissue, where we don't have lymphatic vessels. We have other mechanisms that are involved in waste clearance. One of them is actually dependent on the blood vasculature itself, where there's a lot of exchange with, your blood circulatory system, okay? Another, for instance, is not really at the level of the blood vessel itself, but it's happening next to the vessel, where you have brain fluids, right, that enter the brain and then go up and meet whatever you have in terms of fluid that are closer to the barriers. And when I say the barrier here, I'm mentioning, for instance, the meninges, which is the anatomical location where the lymphatics are, right? So what was really, really interesting is that what we thought to be a clearance system that was somehow closed because you have fluid circulating within the brain and then being excreted outside of the brain tissue itself. But then it was kind of, it was, for some people it was weird, because then where does it go, right? You have this fluid forming, and you reach these compartments within the central nervous system, and where does it go? And what we saw is that, okay, now we have another potential explanation for fluid renewal and fluid outflow from the brain environment, outside of the brain environment, because you actually have lymphatics that are present at the borders that then can drain those fluids and those molecules out, connecting what we consider to be the brain lymphatic system to the peripheral lymphatic system. So now we have an actual bona fide vasculature, the lymphatics of the meninges, that are draining stuff out of the brain. That's probably playing a major role in what you were mentioning, right, in terms of waste clearance and renewal, because you have cerebrospinal fluid being formed at particular parts of the brain, especially by this tissue that is called choroid plexus. That's the tissue that actually secretes a lot of these ions and transports a lot of these ions that form the fluid of the brain. This fluid usually it washes the brain. Okay, this is not a very scientific term, but it's washing the brain, and then you have these and through these pathways that I'd mentioned, right? These vascular associated paths, and then you have the lymphatics at the border that are also draining fluid and molecules out. Okay, so I think we have now a more complete picture. Let's put it like that. However, there's still a lot of details that we don't know, right? A lot of contribution. What is the contribution of the lymphatics that are present at the borders for the total waste clearance of the brain, right? Because you have all of these different pathways. Is it responsible for half of the waste clearance? Is it responsible for 25%, right? So we don't have a number. We know that it does that. It's involved in that process, but we're trying to figure out the exact contribution, right? And then again, we're talking about a very particular type of vasculature, right? The lymphatics are they have their own characteristics, right? And as I mentioned in the beginning, these vessels, they develop postnatally, right? Which is something very particular when you're talking about lymphatic development. So there must be some other aspects potentially that are characteristic of the meningeal lymphatics. And we're trying to understand, okay, what are those? Right? Can we modulate them somehow? But again, I feel like we're just seeing the tip of the iceberg at this point. We need to continue studying it and at all different levels, basic science and also do more translational science.
So one of the things you said during BAMF was, you talked about the meningeal lymphatics drainage into the deep cervicals.
Yes.
I talk a lot about that too. And I'm a big proponent. I come from the clinical side, right? Lymphedema therapist, working in people's lymphatic system. And I'm always encouraging people to massage their own terminus, right?
The neck area.
The neck area. So it's not a big jump to say to people, this is beneficial to massage your cervical lymph nodes, correct?
Potentially. I mean, I listen, I live on facts, right?
I know.
I'm being a scientist. I always like, okay, show me the experiment, show me the results, and then it's going to be. So can I tell you, yes, it's definitely beneficial. I won't tell you that, but if based on what we know right now, right, and that these lymphatics, they drain to these cervical lymph nodes at the level of the neck, and what we know is that in human lymphatic at the area of the neck, you have a lot of cervical lymph nodes. You have a lot of lymph nodes that drain the face. Alongside with your central nervous system, with the brain in this case, okay? So if I would have to guess, which I usually don't like to do, right? But if I would have to guess, yes, I would say that stimulating lymphatic at the level of the neck will probably be beneficial, right? Because it will lymphatics, that's how they work. They need this pulsatile, contractile motion to promote the unidirectional flow of lymph. So if I would have to guess, I would say yes. It's probably going to help. It's going to help,
right? So, Ian Scholes, who was the certified lymphedema therapist who opened the conference in Banff, he's a certified lymphedema therapist. One of the things he said was, we, as clinicians, we can't wait for the science to catch up with what we're doing.
Right.
Using the application of your research and your knowledge of anatomy and physiology, and we're applying it, and we are seeing benefits in our patients. So just throwing that in there. Now, I'm wondering, do we need a description for our audience about what is the meningeal lymphatics? Can you just tell us what that is briefly so that everyone's on the page and they're not looking it up on their iPhones right now?
Absolutely. So in very, very brief words, so you have your brain tissue, right? Your brain. Think about the brain structure as a whole with all the neurons and all that. So you have this tissue layers that surround the brain, okay? And they're there to promote, a proper intracranial pressure, right? The pressure that your brain needs to maintain its a division between what is considered the brain and the outside and the peripheral system, right? So in this tissue that is around your brain, okay, you have a lot of different cells and you have a lot of vascular structures. We knew that you have a lot of blood vessels. What we found out is that you also have lymphatic vessels in those structures that are around your brain, okay? And these are actually not only present around the brain itself, but also the spinal cord, right? So in the spinal cord, you also have these layer of tissue that surrounds the central nervous system tissue. And across those tissues that wrap up your brain, you also have lymphatic vessels. And these, again, in a process that we're still trying to understand, have access to brain-derived content, either fluids or molecules. And that's the part that we're trying to figure out, how do these molecules reach the lymphatics? What is the full anatomy? And again, it's a bit easier to understand in small mammalian species that we use to, as models. But I think it's going to be very important to understand the human physiology as well, right?
So then how does that differ from what we've all come to learn is the lymphatic system with the glial cells in the lymphatic system? Can you explain that for us?
Yes. So the lymphatic is part of the clearance mechanisms at the level of the brain itself that I was talking about, right? Those spaces that you have between your blood vessels of your brain, right? And the rest of the brain tissue, and the lymphatics, the G comes from the glia, right? The glia forms with its a part of the cell that is called end feet. It forms a barrier, a sort of barrier between what is fluid and the blood vessel. Okay, so if something comes from the blood vessel, right? And for some reason, you have particular pathological conditions and you have blood content that is trying to, come into the brain, usually that's not a good sign. And so the glia and feet are there also to prevent, it's another form of barrier to prevent access to the brain. But what's fantastic, and again, also groundbreaking work from Mike and Nedergaard's lab showed is that between that glial end feet and the blood vessels, you have space through which fluid can circulate as well. And so that's one of those mechanisms at the level of the brain itself that promotes fluid exchange, okay? But again, what we work that Dr. Kipnis started doing, and then later on, I also, as a postdoc in his lab, and now in my own lab, what we're trying to figure out is then you have this fluid exchange in the brain. How does the fluid get out?
Right. And that's through the meningeal
lymphatic system.
Clearance.
Yes.
Okay. Thank you for breaking that down for us. Now, the other thing you were mentioning about, which I want to dig a little deeper into, is the cerebral spinal fluid and the central nervous system and how lymphatic inflammation and the nervous system disorders are connected, related. How can that drainage system benefit central nervous system disorders, et cetera, go?
Yes. Yeah. That's the $1 million question, right?
Yes, that's why you're on.
Right. So that is actually an aspect that we're trying to understand in my lab right now. It's, and we're using gain and loss of function models in terms of lymphatic drainage. And then we're looking, okay, if you increase lymphatic drainage at the level of the meninges, or if you cut it down, okay, what is changing in the brain? Okay, because we that's the thing, we don't really know, right? Initial studies that we performed, we know that for instance, the rodent behavior, how little mouse behaves if you impair lymphatic drainage, right? And we've shown that it's actually, they behave differently. And your behavior usually goes hand in hand with your brain function, right? If you have a headache or if you have some sort of infection, your brain responds to it and it alters your behavior drastically. And what we've shown is that if you dampen meningeal lymphatic drainage, the same behavior is affected. And this is completely related with how your brain is working. But we don't know if there's a particular cell in your brain that is dysregulated because of lymphatic malfunction, right? We don't know if it's all cells in your brain that start to, respond due to poor lymphatic drainage. And this is kind of, now we're using different experimental paradigms to try to tease this apart. Okay, is the entire brain as a whole tissue falling apart? Does it start with a particular cell type that is very sensitive to changes in brain fluid content, because you dampen this outflow of fluid through the lymphatic system? And that particular cell will then dictate how brain will respond further on. Is it just molecular content in the brain, right? Because you're draining fluids, but you're also draining molecules, right? You're promoting this waste clearance that we're talking about. Is it a more like a general response? Because molecular content of the brain is being altered, right? And obviously in any tissue, right? Edema, right? Edema is accumulation of fluids, accumulation of waste, accumulation of molecular content that you want to get rid of that in that tissue, right? And so the brain, we think it might be similar, but we need to understand, because again, brain is a unique tissue, right? When you think about it. Extremely complex, right? Extremely complex. It has to be fine-tuned, right? If there's some change in your brain, I mean, small changes might have huge implications, right? And so that's where we're trying to understand what is lymphatic system doing to this fine-tuning of brain function, right?
Right, and the importance of that. Like, I think about that with mild concussions.
Absolutely.
Right? Or like, sometimes fluid comes to the brain for healing. And then what's that clearance? And I do so appreciate that now we're aware that the lymphatic system is apparent in the clearance of it to promote. And that is an important aspect that I think people haven't been talking about in these instances.
Absolutely. And there's more and more work, again, more at the level of basic research, and using a lot of, different models, showing that you can actually, lymphatic drainage is involved in resolution of brain edema, right? Accumulation of fluid in the brain. And I know because, I'm closely involved within this field. And I even if the studies are not published, I see the studies, I discuss with scientists that are doing the work. And again, people are asking, I feel like in general as a community, we're asking the right questions and we're getting to that answer, right? It is a very important system to maintain brain fluid homeostasis. An aspect that we didn't really delve much into is also, it's very important to modulate inflammation in the brain. And when I say inflammation, it's not even the bad inflammation, right? Because in the brain, you also have these immune cells that are necessary for the brain to function, right? They're helping the neurons to fire, right? And they're there to support them in a way. And we're realizing that, because lymphatics, they also have this very tight relationship with the immune system.
That was going to be one of my next questions. Yes. Talk to us about that.
Yes, because, for instance, and it's very easy to understand when you think about infection, right? When you have an infection, the immune response against that particular infectious agent, it starts in your lymphatic system, right? It starts in your lymph nodes. That's where the immune cells are, and they respond to all of these antigens. Well, the antigens are pieces of those infectious agents that are then going to be recognized by your immune system. And the brain draining lymphatics, we believe that they're going to act the same way, right? They're going to be there also to modulate the immune system within the brain environment, whether you have an infection or just in normal conditions, right? Because if you think about it, if you have less drainage, you're also going to have a different immune response. When you have a different immune response, it's going to affect the tissue, okay? And again, but these are those small aspects that we're still trying to figure out, right? Trying to better understand.
Thank you for that description. I think it's just such gold for people to really understand that role the lymphatics play. When you were talking about cognitive behavior and the role of lymphatics in rodents, I know that one of the studies at the KIPNIS that you might have been a part of was how they noticed that in rodents with the narrow lymphatics, they were having a hard time getting out of the maze. Whereas before they could get out of the maze, and once there was lymphatic impairment, they could no longer remember how to get out of the maze.
Yes, right? Absolutely. Yes.
Now, I don't know if this is published or unpublished or not, but at the Lymphatic Forum, Susanna Fabri, she was also mentioning stroke victims, right? And how it recruits migratory VEGF-C producing cells and the leukocytes and adhesions. And I don't know if that's something we're allowed to talk about, but can you talk to us and about what's going on with stroke victims and the lymphatic system?
Well, I mean, what I'm going to say again is based on studies performed in mammalian systems, not in humans, okay? But yeah, there's studies out there, more than one actually, that are showing that if you have, and again, you mentioned also concussion, right? We can model that in animal models. We call it traumatic brain injury models, and also in stroke models. You usually have accumulation of fluids, right? During that, because there's an impact, there's tissue damage, and usually there's a response to try to cope with that damage. And there's usually blood vessels that are ruptured. And so there's usually accumulation, most in the acute phases of those injuries, there's accumulation of fluid. And there's usually immune cells that are recruited to those sites as well to try to clean up things, right? Because there are cells dying, they need to clean it up, they need to resolve it, bring the system back to balance. And again, when you think about draining excessive fluid, right? Regulating the immune response, what comes up to mind, right? The lymphatic system, right? The lymphatic system. And what these studies have shown, again, without going much into detail, which I think it's what we need to do more and more, is that if you have impaired meningeal lymphatic system, the outcome in these models, right? The traumatic brain injury or the stroke is going to be worse. You're going to have a worse outcome because it's going to take you longer to recover from these insults. Now, again, if you ask me, okay, if we're talking about people, right, that had a stroke, or they had these repeated concussions, or even a harder concussion, right? Do they need to increase their lymphatic drainage of the brain? If I would have to guess, I would, based on what we know right now, I was going to say yes. However,
drainage.
Exactly, promote it. The thing is that we have to be very careful, right? Because most of the ways that we use to modulate lymphatic drainage in a particular context, right, that we think is going to be beneficial, if it's prolonged in time, or if there's some other underlying condition that we're not aware of, right, it might not be as beneficial. So I feel like right now we're still in that discovery process to understand, okay, how can we modulate the system, first of all, right? And let's say that, you had a concussion, we need to increase your lymphatic drainage, right, of the brain, but you have also this underlying condition. We only have to do this for a limited amount of time, and then we have to stop, because otherwise, if you have too much lymphatic drainage, right, it might also be detrimental because you also have this other underlying condition, and we don't want to increase drainage of brain content into the peripheral lymphatic system. Because, for instance, what we think, and again based on basic research studies, is that in autoimmune...
Yes. Oh, yes.
So autoimmune responses in the brain, you do not want to promote, right, in the long term, drainage of brain antigens, because that's what's driving this autoimmune. So basically your immune cells to recognize brain antigens and to go there and attack your brain, right? And this is what characterizes, for instance, multiple sclerosis. And so what we're trying to figure out, another aspect amongst the many aspects that we're trying to figure out, right, is, okay, there's a particular response in this disease. Do we want more drainage? Do we want less drainage, right?
So that's what I'm always talking about, is the sweet spot.
Yes.
What is that sweet spot of lymphatic drainage? And I'm always also talking about, there's many different methods of lymphatic drainage now. And, the way I studied the Vodder method is it's very, very light. And we are not draining. We're working within the cadence of the lymphatic system. We're working slowly and we're working gently. We are not overstimulating the lymphatic system. We don't want to work hard and fast and long, right? Slow and steady wins the race. And that's what I'm always talking about. And even I work with people with autoimmune conditions, but we do it in a very controlled small manner, right? And so that I'm always getting people to understand, to find that sweet spot of drainage where you're not overdoing, but you are doing just enough to help encourage that lymphatic movement.
No, I totally agree. I mean, that's what we have to figure out, right? And I feel like it has to be a combination of different studies, right? Studies at more molecular and cellular level, right? And we use that, animal models or in vitro, so cell culture models, right? Usually to find that these more mechanistic. But we also have to understand more at the level of, anatomy, right? Like big tissue level. What is the contribution of lymphatics? And how can we then combine different approaches, right? Maybe what we have to do is find that sweet spot that you're doing in terms of manipulation of the tissue and of lymphatic drainage, plus giving the person this, compound that is going to, at the molecular level, also make your job a bit easier, right?
Correct. And I also say, let's wait to start. I don't start lymphatics right away. Let the body do its job. So the other thing I want to ask you about is, when we were in Banff, you were also talking to us about lacteals and the role of lymph in the small intestine related to the brain and the fat lipids. And so can you dig into that for us?
Yeah. So one thing that, and that was I use that as a sort of analogy, because one hypothesis that we're putting in the table and we're testing it is that we think that the brain is composed, it has a huge lipid component, okay? Half of your brain is made of fat. Let's put it like that. It's a lot of water and a lot of fat. And so the lacteals, so the lymphatics of your small intestine, their main function is to absorb dietary lipids. Okay, that's what they do, right? You have a bunch of different lipids that comes from what you're eating, and they're there to absorb the lipids, because you need some of the lipids, as you need another, bunch of other nutrients. And what we think is, I was using as an analogy, we know that the lacteals, which have been more studied than the lymphatics that drain the brain, they're involved in, modulating this lipid reabsorption. So the hypothesis here, since the brain has a huge lipid content, we think that the lymphatics that are draining it, they're probably also involved in modulation of lipid content. And so, we're devising a series of experiments to test this, and also not only to test it, but also to understand the implications. Again, right, because now we have some data showing that it affects lipid composition in the brain, but how does it do it, right? Is it affecting a particular cell response, right? Is it the cells that actually are involved in forming these lipid-rich structures in the brain, right? Is it some other cell? Is it an indirect effect? And so we're going a bit more into detail, but yeah, so we think that the meningeal lymphatics are kind of the lacteals of the brain.
Got it. Because, there's all this, in the health and wellness world, there's all this talk about gut-brain axis.
Yes.
So, it makes sense that the lymphatics are involved in that as well. We know they're involved, but it's just a matter of to what detail. Okay, couple last questions before I let you go back to your lab. The other thing that Dr. Fabry was talking about was she was talking about COVID and the central nervous system invasion through the cribriform plate.
Yes.
And how smelling is a common issue in COVID patients as well as Alzheimer's patients. So can you shed some light on the lymphatic role that maybe you have seen in that area? So,
COVID is a respiratory virus, right? And so what we have is this structure that you were just mentioning, the cribriform plate. It's very hard to even for me, and I say it, and I say often, I can tell you. And so this structure, it's formed by bones, but they have porous, so they have holes in it, and these holes is where your olfactory nerves go through. Okay, and so you have this structure which is a little bit more permissive, let's say like that, right? And so you also have a brain content, fluids and molecules that can, in particular conditions, through these structures, access a bundle of lymphatics that are present outside of the central nervous system, which we call in the field nasal lymphatics. And these nasal lymphatics, they're also draining into the cervical lymph nodes, right? So back to those, exactly, at the level of the neck. So they're draining, this part around the cribriform plate, although they're considered already outside of the CNS, they do have access, right? And then you have also the meningeal lymphatics, which are embedded within those tissues around the brain, right, that I told you about. And so what Dr. Fabry is saying, again, if you think about, viral infection, you need to fight it through the immune system. And what we think is potentially if the virus, COVID virus reaches the brain, right? And there's a lot of people, they were talking about brain fog, right? And all of these different symptoms, that have to do with your brain and how your brain works.
Long COVID, right.
Long COVID, exactly, right? And people, they're saying, I cannot, work the same way. I cannot pay attention the same way. So these are all brain-related symptoms. And what Dr. Fabry was saying is that, potentially, who knows, alteration of lymphatic drainage at these spots, right, the nasal lymphatics, the meningeal lymphatics, might be involved in the development of brain COVID infection or in the development of these side effects from COVID infection, right? Because again, we're talking about a type of vasculature that is super involved in modulating immune responses, right? Super involved, at least in the periphery, in modulating, response to infection. And so again, it's one of those other areas of research that might be super interesting to delve a bit deeper.
For the listeners, that's why myself and other lymphatic practitioners have worked with people with long COVID, or once they're not in the infected stage anymore, it can be very beneficial for people with brain fog, doing some of the applied manual lymphatic drainage techniques. So starting to close this conversation, which again, I could go on for another hour. So in terms of therapeutic interventions, are there any approaches that target the meningeal lymphatic system that you're seeing results or you're seeing promise down the road?
Well, we started doing, the modulation of meningeal lymphatic system based on what we know in terms of peripheral lymphatic biology. And so there's one growth factor. It's called vascular growth factor C, vascular endothelial growth factor C, VGF-C. Okay,
right. That's during the lymphatic form. Everyone was like VGF-C.
Exactly. So that is kind of the growth factor for lymphatics. It's super involved in their, at least in the meninges, in their maintenance, according to what we know right now. And so that could be potential, therapeutic agent that we could administer in certain conditions. So for instance, we've shown that in a very, very old mouse, if you give them VGF-C locally at the level of the meninges.
And what does that look like? An injection or a pill? What is that method?
Well, right now, so what we did in the mouse, and actually we used the strategy that was used in the COVID vaccination as well. We have this, what is called adeno-associated virus, that can be injected, and then it basically produces whatever peptide, whatever small molecule you want, right? And for vaccines, that one of the strategies was this one. So you have a viral construct that is expressing the peptide that you want to form that immune response against. So it was, in the case of COVID vaccination, it was expressing one of the molecules that is expressed by the virus, right? And then it gives you long-term protection. Very briefly, we use this strategy to deliver the VGF-C into a very, very old mouse. And what it resulted is that the lymphatics in the meninges started to drain a bit better. And the mouse, the very, very old mouse, which had a hard time in that cognitive task that you just mentioned, it started performing better, okay? And so is this, the thing, right? Should we start, treating older patients that have these cognitive impairments with VGF-C? I don't want to go that far, right? I don't want to go that far because it's too early, right? Because we did experiments in a very simple mammalian system. We are much more complex, right? We live longer, right? There's a lot of other factors it might be promising, right? I'm not saying otherwise. But is this going to be the thing? At this point, we cannot say, but we have to do more experiments, right? And again, do a little bit more of those translational studies that I was telling you about, right? Maybe, starting to go into human condition, develop smaller clinical trials where we can test these drugs, right, that show promising results in more simple mammalian systems like the mouse, for instance, and then we can see the outcome. Okay, do we get an improvement in certain condition, for instance, like Alzheimer's patients that have memory impairment? If we give them VEGF-C, and sometimes might be VEGF-C, as I was saying, plus something else, right? Right? It might be a combination. It might act together synergistically with something else, right? And then you achieve that end goal that you want, which is to improve people's lifestyle, basically.
I imagine you'd have a line out the door of parents of Alzheimer's patients signing their parents up to try it, because if you've ever been with anyone who's had Alzheimer's, it's just
devastating.
It's devastating. It's really...
It is one of those conditions. All of these aging-related neurodegenerative disorders, they're very tough on the families. So
right, but that also gets to that. What again is the sweet spot of when you try it? When you're at early stage or mid to late stage, right?
Absolutely.
There's so much there. Okay, coming back to where we started about diet. And food. And I don't know if you've done any research or it's just been, what's your thoughts on what does impair the lymphatic system, diet-wise, health-wise, lifestyle, and any kind of things that you'd say, all right, people, here's my takeaway to benefit your lymph?
I mean, if we're talking about the lymphatic system overall, absolutely. I think diet is probably going to have a huge impact because, again, your lymphatics, they're super committed, right, into, we're just talking about the lacteals. They're super committed into draining all of these lipids, right, all of these nutrients that are arriving through your gastrointestinal tract. And it's the same as the level of the tissues, right? They're reabsorbing a lot of things, a lot of nutrients, a lot of waste products from the tissues, right? And I feel like, if you're having a very unhealthy lifestyle, right, eating all sorts of junk food and, it's definitely going to impact your lymphatic system because you're gonna give it a lot more work, right? Too much work potentially at a certain level where it stops doing what it's supposed to be doing, right? And is only trying to catch up, right? Catch up. So you don't want to overload your lymphatic system in a way. And there's everyday things that you can change to maintain your lymphatic doing its normal job and preserving your tissue function, right? Your gut function, your heart, because for instance, in the heart, you also have lymphatics that are very, very important, right? And of course, obviously I'm biased, but your brain, right? Your brain, you want your lymphatics that drain your brain to be there and maintaining all of that activity that you have all the time. And so I feel like lifestyle and diet is a huge part of it. I think definitely having a good diet, a good lifestyle will be super helpful. If you have that, right? Plus, right? If let's say you have a disease that is familial, right? No way around it. It's in your genes. You cannot do anything about it. Having a good lifestyle plus taking, that drug, that compound that's going to help you, it's always gonna work synergistically, right?
That compound's not available yet.
It's not available. It's
coming. So, right, diet, lifestyle, exercise, stress, sleep, the whole anti-inflammatory bucket
I agree.
Not overwhelm the lymphatic system is what we're all about. And doing lymphatic massage, y'all.
Absolutely.
And the neck, everywhere. I mean, I'm a big fan of doing lymphatics everywhere and in the gut, in the abdomen.
Yes, I have to start doing a little bit of, a bit more neck massage to also fire. Absolutely. Please do that.
All right. So this is the segment where we do our in and out of flow, kind of like the bookend of the icebreaker questions. To recap, I talk a lot about being in and out of flow, right? In a state of flow. So what does being in flow feel like for you? Do you ever get to have that experience?
In flow. Yeah, I feel like, when there's a lot of things that align, professionally, personally, I feel like, that's when I feel I'm in flow. Everything's coming together, no stress involved, right? Which is, for people doing our job is it can get a little bit overwhelming sometimes, because we have to deal with all sorts of life events, with people, with experiments, with conference, with travel, we talk, we interact, we network a lot, right? And it's part of the job, but sometimes, right? So I would say my inflow moment is when everything kind of aligns professionally and personally, and it's for the better of everybody, I guess, for me included.
Yes, I love that so much. And like meeting in the elevator. And this is one of the things I said to you when we were in Banff. I said, I love that you're you have your own lab that is part of the Mayo Clinic in Florida, and that you get to be, the jefe, the man now who's like, yes, you have your researchers, you have people that have to provide your research for, but that you get to create.
Absolutely. Yes. And I really enjoy, working with younger people, and together as a team kind of dictate where we think the science should lead us, and, tackle those very, very tough questions, having discussions. I think that's also a very rewarding part of having your own group and starting your own lab. And I mean, it's challenging, right? I'm not going to say it's not challenging. It's a different type of challenge, but it's very rewarding, right? I think that's probably, if I would have to name the best part of being a group leader, I think that's it. It's working with fantastic crew and trying to, do the best science for the benefit of the people.
I love it. Thank you so much for being on. How do we find you? Are you findable? How do we keep up with you and your research and what you're doing?
Well, I mean, if you go to Mayo Clinic website, and you find, probably if you Google my name, you're going to find our lab website. You can see all the amazing people that are working with me right now. You can see, in broad terms, what those questions that we're trying to tackle are, and if you want to reach out, we're always available by email. So it's very easy to talk with us if you want to know more about our science, or if you just want to know a bit more about ourselves, right?
and you were just at the Alzheimer's Association convention. You said in Amsterdam. You're available if somebody wants you to speak.
Absolutely. Yes. I'm always available to talk with people, to do presentations, talk about our work, and just have discussions. And now with Zoom, I guess that's one of the benefits, right? Even if we're a world apart, we can always, get together.
Well, thank you so much for taking the time today. I've loved our chat. I know our listeners will have loved this too. So everyone, do your lymphatic massage and keep thinking about the drainage and the beautiful health of your brain.